Before I get started, I'd like to begin by acknowledging the traditional owners and custodians of the land on which we meet today, the Wurundjeri people of the Kulin Nation, and pay my respects to their elders past, present, and the elders from other communities who may be present today. It's my great pleasure to introduce our speaker today at the Wednesday seminar, Jinjin Chen. Jinjin's been a PhD student in the Davis Laboratory here at WeHi for a number of years. She started her PhD back in 2020 during the middle of the COVID pandemic and spent the first year working remotely from her parents' lounge room in China, which really demonstrates the dedication that Jinjin had to get started, get going, and start doing the science that she's going to present to you today. She did her undergraduate degree at Nanjing University and was actually part of our Inspire program here many years ago, really indicating just how fabulous that program's been for recruiting top students to the Institute. As I said, she started her PhD remotely in China, and then we managed to get her into the country during one of the brief breaks where we were able to get students in. So she was obviously working on our COVID project, looking at spatial transcriptomics in COVID-19. And so that really spurred an interest for Jinjin in working with spatial transcriptomics data at single-cell scale. She started off looking at cell type identification and deconvolution in cancer, particularly colorectal cancer, but pretty quickly started focusing in on these problems around how to identify different cell types in new data sets like spatial transcriptomics. So she's been able to apply herself absolutely brilliantly to this task. Jinjin has been a fabulous student and very creative along with hardworking. She's done an amazing amount of work over the course of her PhD, and I think it's going to be really interesting for you all to be able to listen to what she's presenting today. So Jinjin, it's a pleasure to introduce you for your completion seminar as well as the Wednesday seminar today, and good luck.
Sorry. Yeah. Thank you for Melissa's beautiful introduction to me, and thank you everyone for attending today's Wednesday seminar, which is also my PhD completion seminar. So today I present the work I've done during my PhD, which is mainly about methods development for marker identification and cell type annotation on transcriptomic data. And I do believe this work would be quite helpful to unravel the cellular and tissue heterogeneity. So, oh, sorry, I forgot to laser. Okay. Yeah, so today I will first give you a brief introduction of the background about the motivation of my PhD and what my PhD aims are. And then I'll introduce more details of my 3 PhD projects and end my talk with some acknowledgement. So first, we need to figure out what is marker gene and why we need to identify them. So marker gene on transcriptomic data refers to a gene which is specific to a group or condition of interest in biological and clinical research. So marker genes can be correlated with different disease status, different treatment response, or they can also be related to different cell type or survival outcome. By identifying these marker genes on transcriptomic data, it can help us to better develop diagnostic tool. And some markers with prognostic value can also help us to predict treatment response and the patient outcome. While some markers might also be the potential therapeutic target, which can help in advance our precision and personalized treatment. Those markers which are associated with disease disease mechanism can also be helpful in monitoring the patient's disease progression and thus further improve the patient management. So all of these good applications of marker genes on transcriptomic data explain why we want to identify these markers. And one general RNA sequencing technology in transcriptomic data is bulk RNA sequencing data, with hundreds to thousands of cells within each sample. However, because bulk RNA sequencing data is a collection of many cells, which may be mixed with more than one cell type, so it cannot give us a single cell resolution, which is quite important for our understanding in tissue microenvironment. For example, here is a tumor microenvironment diagram, and we can see there are very different cell types, which each performing specific functions. And only by understanding the expression pattern on each single cell and across different cell types, it can help us better understand how different immune cell types can respond to different disease or immune response. And how different tumor cells and cancer-associated fibroblasts can contribute to the disease progression. So then we have single-arm sequencing data, which can help us to realize this point. So with whole transcriptome in each single cell, now we can better understand how different cell types can, what kind of important roles of different cell types can play in different disease, and how different pathways can be modulated in different biological processes. So this can give us a better understanding of the tissue heterogeneity. But we researchers are greedy, so we won't stop here. With technology advances, now we can even have spatial single-cell data, which can provide us not only transcriptomic data at single-cell resolution, but also for each single-cell spatial location. And with this new technology, now we can even explore how different cells can interact with different cell types in its neighborhood. And this can definitely provide us a more comprehensive cellular landscape across different tissues and organs. So all of these technology are quite helpful. However, with technological advances, a lot of limitations also come out. For example, sometimes, depending on the research purpose, single-arm sequencing is not always feasible or preferable. At this situation, bulk sequencing is the choice. However, with the progress and popularity of single-arm sequencing technology, more and more methods are developed only focusing on single-arm sequencing data or other new data types, while ignoring the bulk-arm sequencing data. This would leave a gap in bulk-arm sequencing analysis. And so far, there is no automated de novo marker identification method in this bulk-arm sequencing analysis. Additionally, as what I mentioned before, bulk-arm sequencing data is a collection of many cells. So when you try to apply markers from identify from one data set to another context-specific data set, you might find some markers may have the inherent background expression within those microenvironments. And many methods would directly ignore this situation. So this would even impair the accuracy when you apply markers on independent data set. Single-cell RNA sequencing seems to be able to solve this problem because all the cells are sorted and purified, but it has its own limitation because we found many methods cannot accurately identify markers for those rare cell population, which means those cells with low number across the whole data set. And those methods would also fail on such an imbalanced data with real cell population in it. And finally, for the spatial single-cell technology, like cosmic SMI data or Xenium data, they look fancy. Yeah, they are actually fancy, but given the short time they have been available, there's no flexible and automated annotation workflow for this new data type to help researchers annotate cells. And even now, only a limited studies have explored how this new data type look like how the data quality it can be and how well the existing methods can work on this new data type. So it's all uncertain and unclear. So with these limitations on different scales of transcriptomic technologies, here comes my PhD aims. So the main goal of my PhD aim is to develop user-friendly and novel tools to help fill these gaps in different scales. And the first aim of my PhD is to develop an automated marker identification method for bulk-arm sequencing data analysis. And then I developed MasterR, which is a marker automated screening tool in R. This is an R software package published on Bioconductor. So let's assume a case. When you have a bulk-arm sequencing data in your hand, what a standard analysis workflow might look like. First, you might need to manually collect those gene sets of interest. But the first problem is that you might don't know where to search these gene sets. And even if you know where to get this, it might be hard for you to know how to access or download it. And after collecting these gene sets, you might also need to manually conduct the differential expression analysis, which include the data filtration, normalization, and design matrix generation. And after the DE analysis, you might also need to manually pick up those top genes across different DE comparisons. additional manual work is also needed if your input data is not a single data set, but multiple data sets, which means manual meta-analysis is necessary, which might include the data integration or the result aggregation. So as you can see, there is a lot of manual work involved in a standard marker identification in bulk count sequencing analysis. And all of this just because there's no automated method developed for bulk count sequencing now. And with master R, it can help users to automate all the procedures I mentioned before. So the first thing is master R can allow users to automatically collect those gene sets of interest from publicly available databases, such as PanGLODB or MSigDB. And users' customized gene lists can also be easily merged into this marker pool. And with this marker pool, master R would automatically help users to apply the standard DE analysis. And based on the standard Lima and HR DE results, master R would calculate a rank product score by using a permutation test. And this score can help to automatically select the markers with consistent higher ranks across all the DE comparisons. And another key feature distinguish master R from other methods is that master R can also take into account the background expression. So for example here, if you identify an immune cell type marker from one dataset and try to apply it on a tumor microenvironment dataset, some of the markers you might find they would be highly expressed by tumor cells itself or some other cells within the microenvironment. And at this situation, the higher score or expression cannot indicate the higher infiltration or activity of your target type. And master R, by using a signal-to-noise ratio approach, can remove those markers with this potential background expression on your query dataset. So this would be quite helpful, especially when you want to apply your markers on a context or disease-specific dataset. And finally, master R also provides various visualization functions to help users easily assess the performance of the signature on datasets. So this is how master R looks like. And here I'll show you a key result of master R application on a real biological data. I use master R to identify the NK cell markers from DICE, which is a large human immune cell dataset. And then I applied the derived markers on an independent human immune dataset. And here is a result on the independent dataset. Based on this heat map, each box represents the marker's expression in each sample. And we can see all the derived markers are significantly highly expressed in NK cells compared with other cell types. And the same conclusion, can be drawn when focusing on this marker's rank density plot and the marker's score box plot. Both results reveal our derived NK cell markers is significantly enriched in only NK cells. So this result proved that as a user-friendly and automated method, master R can identify cell type specific markers from bulk count sequencing data with a good performance on independent dataset. And by removing the potential background expression markers, master R can even increase the estimation of the markers on independent and disease-specific dataset. And although I didn't show result here, but I also compared master R-derived NK cell markers with those previously published NK signatures, which has been manually curated. And the result proved master R-derived markers can perform as well as, if not better than those published ones. So this means master R can save a lot of manual work from the experts with achieving quite similar performance. This work has been pushed to bioRxiv as a preprint, and we are working on the submission to the journal now. The package has been available on Bioconductor since last year, and we already got over 1,900 downloads. So we were happy to see if any of you would be interested in using it. So now it seems by using master R, I successfully fill a gap in bulk count sequencing analysis. Then let's move to the marker identification in single-arm sequencing data. Here, to identify the markers for real cell population, I will introduce SmartID, which is a gene set scoring and marker identification method based on TF-IDF. To ensure SmartID be able to identify markers for real cell population, here I will introduce TF-IDF, which is a widely used approach in many aspects, especially in text mining and natural language processing problems. And it is actually composed of two components. One is term frequency, is the frequency of a word within a document. And the other one is inverse document frequency, which is the inverse frequency of a word across the document files. And by combining these two components, TF-IDF has been proved to achieve good performance in identifying the word which is specific to a subset of document files, even if this document files is just a small collection across the whole document files. And this is quite similar to what we try to do on real cell population on single RNA sequencing data. So I employed TF-IDF in our smart ID scoring method, and I also did some modification to make it adapted to single RNA sequencing data by regarding the cell as the document and gene as a word. So with this new score, the scaling is also necessary here because different genes may have different scales of expression. And without a proper scale, the bias would be introduced towards those genes with larger numerical range, and that's not what we want. So here I used a customized mean of group mean scaling method to ensure that each gene can contribute to the importance calculation equally. And this customized scaling method can also handle with those imbalanced label data. Then a series of transformation would be applied on the scaled score. Finally, the score would be converted into an importance with the range from 0 to 1, which would be easier for the feature selection. So after all these steps, the final the distribution of the real markers would look quite different from the known markers. And at this point, I used a Gaussian mixture model to help us automatically identify the distribution of the real markers and then further identify the final markers. So this is how Smart ID looks like. And here, I will show you an application of Smart ID. So here I use Smart ID to identify a CD14 monocyte cell type marker on a public dataset, PBMC3K. And then I use Smart ID to score each single cell using our derived markers. And based on this score box plot, it's clear that Smart ID derived markers can clearly separate our target cell type from all other cell types. And to further assess how well this performance is, I compared it with SURET, which is another popular and widely used analysis tool in single RNA sequencing analysis. And based on the SURET result, we can see, although the target cell type also jumped out, but it would be so difficult to distinguish it from another similar monocyte, which is the FCG monocyte. This means the markers written by SURET actually contain a lot of commonly shared genes between similar cell types, while Smart ID can give you highly specific markers, which is only specific to the target group. To further assess how well Smart ID can perform on the real cell population, I use Smart ID to identify the NK cell markers, which is the smallest population in this dataset with 6% of cells. And similarly, we can see Smart ID actually can get a better separation of NK cells compared with other cell types, while the difference is like smaller when using SURET. And we notice the score level is kind of similar between NK cell and FCG monocyte. And then we realized this FCG monocyte is the second smallest population in this dataset. So this result just proved, compared with SURET, Smart ID can better handle with this real cell population in this marker identification. And then, because Smart ID can actually assign a score for each gene in each cell, so it can also be used as a data transformation method. So here, I run t-SNE by using the standard log normal counts and our Smart ID score. And t-SNE here is a dimension reduction method for better visualization. So with this method, it can help us better visualize how well the cell can be separated. And based on this result, it's clear when using Smart ID score, the similar cell types can be clearly separated. Like here, we can see the CD8 T cell can be separated from CD4, while they would be all merged into one large cluster when using standard normal log accounts. And similar conclusion can be drawn when we focus on these two similar monocytes. So all of these results prove that, first, As marker identification method, Smart ID can accurately identify cell type-specific markers, especially for those rare cell population. And secondly, as a data transformation method, Smart ID can also give a better clustering result compared with standard normalized log counts. And this package has also been available on Bioconductor recently, and we are working on the submission of this manuscript. So if any of you are interested in it, we will be happy to take any feedback. Okay, so now we can move to our final session, which is the spatial single-cell technology. So for this new technology, what I aim to do is to build an automated ensemble annotation workflow to help users annotate cell types on this new data type. And the first thing I want you to notice is that the data I present today is from CosMx as my platform. And different from traditional single-arm sequencing data, this data is not a sequencing-based data, but an image-based data. And that's why it's also called a single molecular imaging technology. So by using the probe with different fluorescence on it, it can detect each single transcript and assign it to each gene. And then the cell boundaries would be estimated by using the cell membrane, H&E density, and the nuclei standing information. And once the cell boundaries were determined, those identified transcripts would be assigned to each single cell. So that's how CosMx as might generate their data set. But based on this technology, some concern might arise because we might get uncertain or inaccurate cell boundaries. For example, here we have a green tumor cell. Well, the green lines are the real cell boundaries, and those black lines are the estimated boundaries. And we can see, under this situation, those transcripts with red circles would be wrongly assigned to this single cell. And this would introduce the confounding expression to this data type, and which can even bring the challenge in identifying the cell type on this data. So far, we have no idea how much this cell segmentation error might be, and don't know how well the existing method can work under such a potential error. So to figure out this, I built such an automated example annotation workflow. So the standard analysis is always to do some quality control check first to remove the cells with quite low quality. And then normalization to be done on this filtered data. Here, I chose GRAN as our normalization method, which is one of the tools in single-cell analysis. The reason is that based on our previous published work done by Dharmesh, we found the library size can actually confound the real biology in spatial transcriptomic data. And GRAN, with less library size effect correction, can actually perform better in spatial domain clustering. That's the reason why we chose GRAN as our normalization method. And then for the annotation methods can be divided into two main categories. One is the reference-based annotation method, which is to find the anchors and project the query dataset to the reference dataset, and then identify the cell type. So for this kind of methods, we need a proper reference with specific formats. And another one is marker-based annotation method, which is to apply a given marker list to the query dataset, and then by using such as scoring method to identify the cell type. So for these methods, we need accurate cell type for each cell type, a marker list for each cell type. And my workflow actually provides functions to help users easily build this reference and marker list from the same reference dataset. Then with all this prepared data, the annotation can be combined by using a majority voting strategy. And this strategy can be summarized simply in this diagram. So for each unassigned cell, it will be finally labeled as the type with the highest votes across different methods. And we also provide an additional module called Relabel on Cluster to help improve the coverage of assigned cells by utilizing the clustering results. And I'll explain more details for this section in the subsequent slides. So after all these steps, if we can get enough well-annotated cells, we will be happy for the subsequent analysis. However, if the answer is no, we will just send data back to the first step for further QC check or filtration. So today, the data I'll present is a COVID-19 Cosmics as my data provided by Aruda from UQ. So thank Aruda for providing this data set and contributing to my project. This data was sampled from 2 COVID-19 infected lung TMA, the tissue microarray, with each TMA was sequentially sampled to generate the replicas, and 2 fields of view was captured per core in most of cases. So this is the overview of the data set. And another thing we need to notice is that Cosmics as my data only provided 960 genes which can be detected. So the Cosmics as my data is not a whole transcriptomic data, but with a selected panel. And they also provide 20 negative probe genes as, for our negative control. So with this data set, the first step is to generate proper reference for the reference-based annotation methods. And here I chose HLCA, the Human Lung Cell Atlas, as the reference data set. Because HLCA is a single-arm sequencing data with whole transcriptome. So to make the result more reliable, I only subset the 952 commonly shared genes between the query and the HLCA to subset the final reference data set. And after searching all the annotation levels provided in HLCA, the annotation level 3 with 25 different cell types was finally selected because we believe this can provide us sufficient granularity in the annotations. And then we also need to generate a marker list for marker-based annotation methods. And here, I passed the same reference to CyberSort X, which is a machine learning method that can infer the cell type specific profiles. So, however, the output of CyberSort X is a signature expression matrix look like in the left box. And what I really want to get is a clear marker list look like in the right box. So to convert the result from left to right, I need to figure out for each selected gene, which cell types this gene might be specific to. And those cell types should be those types underlying this blue box. So to automatically identify the types under this blue box, I used HDB scan. HDB scan is a density-based hierarchical clustering method. So what it did is it can build a minimum span tree to connect all the cell types for each selected gene. And then by maximizing this valid area in this condensed tree, it can automatically decide where to cut its edges to form the final cluster and outliers. Based on our assumption, if one gene is specific to 1 or 2 cell type, they should have a distinct density and high expression. So using this method, this LAMP2, which is a well-known macrophage marker, can be successfully assigned to macrophage only. And similar result can be obtained across all the selected markers in our dataset. So with this method, now I can successfully generate highly specific marker lists for each cell type on the same reference dataset. Then, with all this prepared data, I can do the annotation now. Here, I selected 5 different annotation methods. 3 are reference-based and 2 are marker-based for kind of balance. And here is the UMAP result with cells colored by azimuth annotation result. And UMAP is something similar to t-SNE, so just to visualize the cell separation. And the first thing we notice is that, different from single-arm sequencing data, we can see the cells on Cosmics data, they are not well separated into different clusters. But there is only one large body in the middle with a number of small branches sticking out around it. This might indicate the less discriminative power of Cosmics data in different cell types. And this phenomenon is kind of similar across all the 5 annotation methods. Then we look into the ARI, which is a metric of the similarity between 2 labels to compare how different methods can agree with each other. And then we realized that in situ type actually perform the best, actually achieve the highest similarity across all other methods. But overall, the similarity looks not high. So we start to think what might be the reason to cause this low similarity. The first thing we thought is maybe there are some cell types with poorer agreements impair the overall similarity. So then I split the ARI into different cell types respectively. And yes, we do find there are some cell types with much significant higher agreements across different methods. And those cell types are the types with distinct functions, like B cells, macrophage, or mast cells. Or they are the cell types which seems to be specific to lung, like 81 and 82. While those cell types with many subtypes in our annotation level, like we have a lot of different lymphatic EC cells, or the cell type with irregular shape, like dendritic cell, they are less prone to agree with each other by different methods. So this result suggests us that the cell types with more distinct functions and the regular shapes would be easier to identify by different methods, and those cell types identification might be more reliable for the subsequent analysis. Then, by using our majority voting strategy, I can combine the strength of all 5 selected methods. And here, I chose the annotation. I chose the votes level 3 as our final annotation result. The annotation level here means the minimum number of methods agree on the same cell type. So votes 3 means at least 3 out of 5 methods agree on the final cell label. So that would be over half agreements. And under this vote level, we can actually get around 56% of well-annotated cells. And among these 56% cells, there are another 11% around cells with full agreements, which means all methods agree. And unexpectedly, we found all these, most of these full agreed cell types are those cell types we mentioned in previous slides. They are 82, macrophage, or B cells. So just further validate these cell types' annotation reliability. So with this annotation result, the first thing I did is map it back to the spatial slide to see how it may look like. So here is the result of core 21 in TMAA8. And we can see there is a clear spatial structure which composed of multiciliated cells in it and basal cells on the outer part. And this result actually corresponds to what we can see from a composite immunofluorescence image, which is a fact we can observe. So this looks like a broadcast structure. And this detailed structure cannot be easily obtained if you are using a cluster-based annotation method, because I did cluster the cells using the clustering method. And even with a high resolution, the cluster method would tend to cluster these bilayer cells into one cluster. So this is another advantage of our annotation workflow. However, if we focus on those unassigned cells under this vote level 3, we found there are still a large number of unassigned cells with vote level 2, which are quite close to the final agreements. So here, I developed an optional module called Relabel on Cluster to try to improve the coverage of the cell assignments. And what I did is, first, I can use the customized cluster method to cluster those highly similar cells into the same cluster. And the best clustering result can be selected by comparing the cluster performance metric. And with the final selected clustering results, we can do the relabel. The first step is to find the cluster with gray unassigned cells in it. So here, the gray points are those unassigned cells. And for example, here, these green and blue clusters, there seems to be no unassigned cells in it. So we can directly ignore these 2 clusters. And then, for all the rest cluster with gray unassigned cells in it, I will try to identify if there is a dominant cell type in the cluster. So here, this cluster, although there are unassigned cells, but there is no one dominant cell type in this cluster. And this cluster looks like more than a mixture of multi-cell type. So we are not confident to assign those gray points to any of them. So for this cluster, we will also keep it as a regional label. And also for this bottom cluster, because the dominant cell type is unassigned itself, so of course, we will also leave it as a regional. And finally, only this cluster with both gray unassigned cells in it and a dominant cell type would be relabeled. And only those gray unassigned cells would be relabeled as a dominant cell type labeled yellow here. So this is how this module works. And what I did for my data set is I used SURET to cluster the cells. And I tried 10 different resolutions from 1 to 10 and calculate 3 different performance metrics. One is MVI, which is a measure of dissimilarity between the two labels. So lower is better. And the purity score measures the extent of a cluster belongs to a class. So higher is better. While silhouette score is a similarity metric between two labels, so also higher is better. And based on this line plot, we can see the resolution 3.5, which are the red points here, actually achieve the best performance with lowest MVI and the relative high purity and silhouette score. So after relabeling unassigned cells under this selected cluster, we successfully get 4% increased assignment across the whole data set, which means now we can actually get over 60% of well-annotated cells. And again, we found most of these relabeled cells, they are still relabeled as those cell types we mentioned a lot of times. Then with these relabeled results, next step is we try to evaluate how confident or how reliable our annotation could be. So another advantage of our data set is that we have the sequentially sampled replicas, and the cellular composition between the replicas should look quite similar. So here, I did a correlation analysis across all the 4 TMAs, and we can see the correlation of the cellular composition between the replicas are much higher compared with non-replicas. And this correlation is even close to the slide itself comparison. So this just provides us more confidence in our annotation results. But if we focus on the fields of view level, we found there is still a proportion of unassigned cells in each FOV across all the slides. And compared with TMA-A, actually TMA-B exhibit a markedly higher unassigned cell proportion. So that made me think, if we can find a metric that can help us to correlate the data quality as well as the annotation confidence. And the first thing I test is negative probe detection. As I mentioned before, negative probe detection are designed by nanostring itself, and they are designed to not significantly express or spatially correlated. However, when I map this negative probe back to the spatial slide, it's clear that it's not spatially random distributed, but with a distinct spatial cluster pattern. And we can see there are much more negative probe detections around this multiple voice region compared with other regions, which might indicate the poorer data quality of the cells around this region. So, given the fact that negative probe has a spatial distribution, to better capture this spatial distribution, here I devised a new metric called the spatial neighborhood negative probe detection. What I did is I modeled the negative probe detection of each cell's K nearest neighborhood and aggregating those neighborhoods negative probe detection by using an inverse distance weighting method, which means the closer cell would contribute more to the final metric. And by using this new metric, it can actually categorize the noise intensity of the spatial location where the cell is located instead of cell itself. Then I mapped the original detection, the new metric, and our annotation voice level onto the same slide. And we can find there is significant co-localization of the higher negative probe detection and our lower voice level. So this means the negative probe detection cannot just be used as a data quality metric, but can also help predict the annotation confidence. But if we focus on those cells with dark purple points, which are the cells with full agreements, we found on this core, there are only a small number of cells with these full agreements. So what might be the reason to cause these low agreements in our data set? If it's the problem of our data quality or it's a kind of consistent phenomenon on the CosMx as my platform. To figure out this, I applied the same workflow on another public non-small cell lung CosMx as my dataset. And the result actually show the coverage of all methods, the coverage of all voice level are highly similar. And our dataset even has a higher coverage on this voice level 5. So this means these lower agreements is a kind of consistent phenomenon on cosmic SMI data. So what might be the reason for this platform? The first thing we thought of is the smaller gene panel, because cosmic SMI data only have 960 genes can be detected. So to validate this, I applied the same workflow, the same reference, the same marker list to another public COVID-19 cell atlas, but which is a single RNA sequencing data. To make sure the result is reliable, I also subset the same gene panel as the cosmics data. But this time we can find, the coverage are actually much higher across all the voice levels compared with cosmics data. And if we focus on those agreement distribution, we can see most of cells in this atlas actually underlying the voice level 5. While most of cells in cosmics data, they only underline voice level 2 or 3. So this difference proves that the smaller gene panel of cosmics data is not the main reason to cause these lower agreements on cosmics SMI. So further exploration might be needed to further test what might be the driven reason to cause this low quality. And here, the quality I mentioned is not about just the data preparation quality, but also the error maybe caused by like this technology and platform itself, like the cell segmentation error I mentioned. But regardless, my work still successfully built a first flexible automated annotation workflow, which can help users easily annotate cells on this new data type. And by using our annotation workflow, we can still reveal some spatial detailed structure, which can actually still provide us promising and insightful results to help us better understand the cellular landscape in different organs and tissues. So I think this work is still kind of meaningful and helpful. And part of the work has been published on Biomedicine, and we are still working on the submission of the whole project and hope it can come soon with coming weeks or months. Yeah. So finally, let's make a summary. By developing master smart ID and building an annotation workflow, I successfully filled some gaps in different scales of technology on transcriptomic data. And I believe this work can help us better advance the bioinformatics analysis on transcriptomic data. But of course, there's still a lot of future directions that can be updated and optimized in the future. For example, the master application can be extended on not only transcriptomic data, but also other multi-omics data. While for the smart ID, the threshold estimation and the scoring normalization can also be optimized to obtain a better accuracy and to make the score comparable across different gene sets. While for the annotation workflow, it can still be updated to like automating the best classroom result selection or optimize the reliable step to increase the accuracy and coverage, and the exploration for further improve the cell quality might also be necessary in the future. So here are some outputs during my PhD. I've been involved in some publications and also gave some poster and workshop presentation on both national and international conferences, and 2 of my software packages are also available on Bioconductor publicly now. So that's all. Finally, I have to say thank you to all the people involved in my PhD. First, I have to say thank you to all those lovely lab members in Davis' lab and Belinda's lab. I really enjoy every moment, like every inspiring discussion and brainstorm with you brilliant guys. And I also have to say thank you to my 3 supervisors. Their guidance, patience, and mentorship have helped me a lot, especially at the beginning of my scientific journey. And I also have to say thank you to my committee members. They really care for me on every committee meeting, and especially after a specific time, their help means a lot and invaluable to me. And also, thank Belinda, Phipson, the whole lab. Thank you for getting me involved in your meetings and gatherings and celebrations. That really helped me a lot in my last year of PhD. And also thank Mengbo and Li Zhong for helping me solving a lot of mathematical and statistical problems. And thank Ashley for always cheering me up, encouraging me, made me feel I'm a good PhD student. So you're also a good, lovely person. And also thank Gordon as our division head, helped me a lot during dealing with those funding and travel stuff. And Maria, as our division coordinator, she did a really fantastic job, helped me dealing with those travel and conference form system, which I'm sick of. So thank you, Maria. You really saved me from my life. And you are the best coordinator I've ever met. Thank you. Yeah. And finally, I also want to express my gratitude to those collaborators and Dataset Consortium for providing dataset and contributing to my project. And I do appreciate Weihai and Uni for sponsoring my whole PhD candidature. And yeah, sorry, it's not still the last, but I also want to say thank you, take this opportunity to express my gratitude to those incredible individual friends who have been part of my life. And I really cherish every moment spent with you guys. And your constant help and encouragement, made me get through those toughest time, and those occasional study breaks also helped me to recover from the heavy work and study. And based on the bottom two photos, there's all the dishes we made ourselves for our gatherings. So sometimes it also made me confused whether I major in bioinformatics or cooking. Because I think my cooking skills actually get much more improvement compared with my research skills. That's really. So just want to express my gratitude to all you lovely guys. I really appreciate this wonderful time spent with you. And among all the friends, I want to express special thanks to my flatmate GE, who is the PhD student from Vince Lab, apparently. But she's actually my breeder, because she feed me every day, every meal when I just came in Australia and don't know how to cook. So I have to say, without you, I will never complete my PhD because I would have starved to die. So thank you, GE. And although I didn't mention this too much, but I really appreciate you to help me get involved in so many outdoors and social events and activities. Without you, if I leave myself alone, I would never attend so many colorful events. So thank you for making my PhD life even more memorable and colorful. And wish you all the best, and hope you can successfully complete your PhD in coming months. All good? And finally, also I have to say thank you to those people who are not in Australia, but in China, those my lovely family members. As Melissa just said at the beginning, I was enrolled remotely in China during the pandemic. And at that time point, because of the border closed, I was kind of anxious and confused and lost because of their love and support that can sustain me during these tough highs and lows and help me keep going until now. So I really want to thank them for always supporting and backing up me in China. Yes, finally, even the coffee in the restaurant deserve a shout, because they fill my stomach when I don't have time to cook. And those coffee also fueled my many night writing sessions. So, and Melbourne really have good coffee, isn't it? So thank you, coffee and restaurant. Without you, I cannot do that. And that's the last. Thank you. I'll be happy to take any question. Thanks.
That was fabulous, and Jinjin has been a very good PhD student. As you can see, that was a huge amount of work that she's done. So we're happy to take questions. We'll grab some microphones. I think she's left us some time. Question at the back. You have to run the mic.
I'm going to be careful how I ask this.
I think we need a microphone.
Yeah, so far I haven't done benchmarking because for the annotation workflow, it's not like a novel annotation method development, but more like how to employ in the existing method in this new data type. So as I mentioned, the majority voting things is try to combine the strength of different selecting methods. And those 5 selecting methods are actually selected based on some previous benchmarking result, but not on spatial data, but on the traditional single arm sequencing data, which was done by Vika, who is a previous master student in our lab. So after this benchmarking result on traditional single-arm sequencing data, we pick up these 5 methods with high performance and short running time. But so far, we haven't done the benchmarking on the spatial data because now we don't really have the ground truth on spatial single-cell data. So even if we do the benchmarking, we cannot make the conclusion which is better, which is worse, because no ground truth for us to make this conclusion. So sorry. Sorry. Yeah, actually, so far we have because the data set we got from Cosmics is already their processed data. And we actually cannot know how sensitive their technology is to those single transcript. And based on our QC check, we found the depth or the counts of detection for every gene is kind of low. Even for the high abundant genes, you might only get around 5 to 10 counts. So which is quite low compared with the single arm sequencing data. So the depth, the reader depth is still I think is a normal problem, not only just constrained in Cosmics, but for Xenium and all the spatial single technology. Now the depth is still a problem. We cannot get too many counts. Yeah. Yeah. Yeah, that's a really good question. Yeah, I do believe by benchmarking based on technology would be more meaningful to benchmarking with the annotation methods. Yeah, but we need to have enough money to conduct such an experiment because spatial single cell data is quite expensive. So maybe after I found a great grant, I might try to do that. But yeah, that's a good suggestion. Thank you. Yeah. Yeah, actually for this question, I do have asked some immunologists to help me manually check those marker lists I generated from the dataset. And thanks for Joanna Groom for helping me validating those immune cell type markers. So we actually have a validation to confirm that those markers generated by my method is valid and specific markers. So. Yeah. Sorry, do you mean those markers which are expressed like with less understanding? Yeah, like, so if we identify some markers we are not expect to express, first thing is maybe something wrong with identification. Maybe because the accuracy of identification actually largely rely on your your data label. So if your label is wrong, like mislabeled something, this kind of situation might come out. Or another reason is that these unexpected markers are real markers, but just because the technology now is advancing, so now we can actually get higher accuracy, and sometimes the long read sequencing can also provide us more comprehensive results. So this might provide us an opportunity to find those novel markers and to validate whether these novel markers are real markers or they are just like mistake. Actually, we can use some other bioinformatics analysis tool, for example, the Damesh also built a VSE package which can help to investigate what kind of pathways can be involved in those gene sets. And by doing this, we might be able to validate whether these novel markers are involved in the real important and essential pathways and the biological processes, and then can help us to understand whether this is a novel, unexpected marker or it's a mistake.
We should have a look at Greg.
Yeah, we're on slide 5. No, 3 are reference-based and 2 are marker-based. Yeah. Yeah, that's a good question. Yeah, that's also one of the concerns I'm having now, because as you see, when I compare the annotation result with traditional single-arm sequencing atlas, the results are much better using the same method, same workflow, and same reference. So that's why we think we need further exploration. If we have such a spatial single-cell reference, whether we can improve this coverage and whether they can work better. And my guess is yes. If we can have a reference data set just generated from the same platform, we should definitely have a good, much better performance when applying this same workflow. But yeah, we don't have such a reference now. So,
Jinjin, thank you.
Thank you.